Chapter 5 covers the topic of gene linkage and how we can use gene linkage to determine how likely we are to get new combinations of phenotypes, and then also how we can use linkage and recombination to do what we call eukaryotic gene mapping, which is placing genes in specific locations on chromosomes relative to other chromosomes. There are several different ways that we can do mapping. The one we're going to talk about is recombinant mapping. At the end of the chapter, actually the chapter introduction, but the end of the lecture, there's a little bit more information on some other more modern types of mapping. And then our last type of mapping is doing what we call a physical map, which is doing DNA sequencing to find very specific locations of nucleotides and genes. But we're going to focus here on what it means to have linked genes and how linkage affects recombination and new phenotypes that we might get in our offspring. So for a very quick review, in chapter 2 and in chapter 3, we learned about the principles of segregation. So this means that alleles separate during gamete formation. So this means that our homologous chromosomes separate. And they end up in different gametes by the end of meiosis. And remember that each gamete receives one copy of each allele. And this happens because we separate those homologous chromosomes in meiosis 1. We also learned about independent assortment in both chapter 2 and chapter 3. Independent assortment, as it works for meiosis, discusses the relative position of each of our tetrads along the metaphase plate. And we learned that if they line up in different combinations, we get different combinations of entire chromosomes all the way down, and then those different combinations get separated into homologous chromosomes. We also talked about the process of crossing over. Crossing over leads to something called recombination. Recombination is where you have chromosomes where different regions have been recombined. So we saw this happen during crossing over between our tetrads, and then when they separate or segregate into separate into separate cells, we end up with these new recombinant chromosomes. Now, recombination can also refer to recombining of phenotypes. And this is what happens when we actually express those chromosomes that are the result of crossing over. We get new combinations that are combos of each of the parental chromosomes. Likewise, we also have phenotypes that are the parental phenotypes that result from those uncrossed chromosomes. And then finally, just a really quick definition, then we'll get into it a little bit more later. When genes are located close together on the same chromosome, we call them linked. And when you have a group of genes on a chromosome, we call them a linkage group. And this just means a group that happens to be together in any location on a chromosome. So we can think of recombination in two different ways. We can think of it as the shuffling of alleles into new combinations, and that's exactly what we did when we were doing our dihybrid cross. We were recombining our alleles into different possible combinations to prepare for doing a Punnett square of two different genes with two different alleles each on separate chromosomes. Now, when we looked at dihybrid cross, we were looking at independently sorting alleles, which means they are on different chromosomes. So we were drawing this something like this: big chromosome with allele A, a little chromosome with allele B, or something like that. We're looking at all the combinations of each of those chromosomes. And we did this one several times. When we cross our double dihybrid, our dihybrid with double heterozygotes, we always expect to get the same ratio. And that was a 9:3 to 3:1 ratio. And in chapter 4, we talked about some reasons that we might deviate from that expected 9:3 to 3:1. We talked about things like lethal alleles and epistasis, which are different interactions that cause an unexpected allele frequency. Another reason that we might get an allele frequency that differs from this 9:3 to 3:1 is that our genes are actually found on the same chromosome nearby to one another. And what happens here is that these genes are not going to assort independently. They're sort of stuck together. And the only way that they can get unstuck is by crossing over and recombination. So when we make our gametes from this dihybrid cross, we are saying that there is an equal chance of any one of these 4 gametes. So 1/4 of each of our gametes is going to be big A, big B. 1/4 of our gametes is going to be big A, little b, and so on. There's a 1 in 4 chance of each one. When recombination happens, we also have a 1 in 4 chance, but the number of offspring is not going to be what we expect. And we'll talk about what that means in just a few minutes. In the early 1900s, around 1905, a paper was published by 3 scientists named Bateson, Punnett, and Saunders. Now, Punnett is the Punnett of the Punnett squares. All 3 of these individuals were cell biologists, and they were interested in studying heredity. So this is really early on. This is not too long after Gregor Mendel, but Gregor Mendel's work wasn't very widely known, and it wasn't very widely applied or understood. Thomas Hunt Morgan was also working at the same time. This is the fruit fly guy. And shortly after the Bateson, Punnett, and Saunders paper, Thomas Hunt Morgan published his paper demonstrating sex linkage of eye color and demonstrating that genes are found on specific chromosomes. But before Thomas Hunt Morgan showed that, Bateson, Punnett, and Saunders were basically kind of replicating what Gregor Mendel was doing, but they ended up looking at some different characteristics in pea plants, and they did a cross between plants for flower color and pollen size. And so they took 2 parents, purple flower with long pollen grains and red flower with round pollen grains. And just like Mendel, they discovered that purple flowers and long pollen were dominant because everybody in that F1 generation had purple flowers and long pollen grains. And then they did their crosses and they got all 4 of the possibilities. They got the 2 parental possibilities, which are purple flowers with long pollen and red flowers with round pollen. So we call these the parental phenotypes, the ones that are the same phenotype combinations as the parents. And then they saw the other 2, which is exactly what we would expect from a dihybrid. We call these other 2 the recombinants. And the recombinants are any individual whose traits are a mixture of the parents. So we've got purple with the round pollen, and then we've got the long pollen with the red flowers. So those are our 2 recombinants. But they noticed that they did not appear to have independent assortment. If they had independent assortment, they would have expected that 9:3 to 3:1 ratio. And they don't really have that. They basically have what is something like a 10:1 to 1 to 2. Something like that. It's not exact because we would expect, if it was independent assortment, 16 different genotypes and 4 different phenotypes. But this is clearly not a 9:3 to 3:1. So they decided that something must be going on, and these chromosomes were not actually undergoing independent assortment. So they proposed that these traits were coupled. Now we would call this linked now, but coupled means going together. And they didn't have an explanation for this because, remember, we haven't seen Thomas Hunt Morgan's work yet. We don't know about genes being on specific chromosomes, so they didn't have a mechanism for this. But they did recognize, because they were doing statistics and chi-squared analysis, that this was a statistically significant ratio here. It was not the same as that 9:3 to 3:1. And you know, when you do chi-squared analysis, if you don't get a statistically insignificant p-value, then you have to reject your hypothesis. And in this case, the hypothesis was that the genes were assorting independently, and we had a 9:3 to 3:1 ratio. So now we know that the reason that Bates and Punnett and Saunders were seeing this coupling is that genes have specific locations on specific chromosomes. And if we think about human chromosomes, we have 23 different chromosomes, and there are about 22,000 genes in the human genome. It's estimated there are about 22,000. So that's a lot of genes per chromosome. That's upwards of 10,000 genes per, around 10,000 genes per chromosome, give or take, because they're all different sizes and some have more, some have less. And so it absolutely makes sense that some genes are going to be located next to something else. And when they are located next to something else, we call that a linked gene. And linked genes are going to essentially stay together during segregation and independent assortment. But they behave by a particular set of behaviors. And we'll see in just a little bit, but I'm going to tell you for now that genes that are considered close together are going to be on the same side of a chromosome. If they are on the other side of the chromosome, the other side of the centromere, they are considered unlinked. And this has to do with what we call recombination frequencies, and we'll look at that in just a few minutes. So when we talk about linked genes, really all we can say is that they are close together and they are far away from others. Now, the closer two genes are to one another, the more likely they are to stay together and not recombine. So if they're really, really close together, it is less likely that some sort of crossing over event would happen in between them. So we just have A and B placed here. A and B are pretty close together. So the chance that there's going to be a crossing over event right there in the middle with another homologous chromosome is pretty likely compared to the chance that there's going to be crossing over between A and D. And there's less chance between A and B than there is between B and D. And this just really has to do with physical distance. You have to have room for crossing over to occur because this is a structural change in the chromosome. And so you have to have space for the crossing over event to occur. So if two genes are too close together, it is very possible that recombination could never happen in between if they are very close together. So we say that genes on opposite arms are always going to assort independently. Now, we're not saying that the chromosomes come apart, but as far as recombination, we say that they are unlinked, and we're just going to go with that for now until we define why that is. So before we get too far into our lecture, we are going to look at how our textbook writes out chromosome notations. So here we have two homologous chromosomes. And recombination is going to happen during meiosis. Now keep in mind that during meiosis, We're going to replicate our chromosomes. We're going to get identical chromosomes. All right, so to simplify things, our textbook doesn't always draw these sister chromatids. And the reason that they don't draw the sister chromatids is that there's the exact same gene on either side because they are identical copies. So we've got a chromosome that is a sister chromatid with A and B on either side, and then we've got another one with A and B on either side. So our textbook simplifies this in a couple of steps. One, they take away the second chromosome and they just say, well, we're just going to mentally keep track that there is a sister chromatid in there. And on this chromosome, we have A and we have B. And on this chromosome, we're going to keep track that there's a sister chromatid there, and we've got A and we've got B. And then they simplify it even further and say, well, we don't need all these extra chromosomes. We're just going to use one bar to represent all of those chromosomes. And on one set of homologs, we've got big A and big B. On the other set, we've got little a, little b. So if we see this notation here, it just means that the original parent chromosomes were big A, big B, and little a, little b. And then sometimes, just to be even more lazy, they just write it as just the letters. One chromosome has A, B, the other chromosome, the homologous chromosome, has little a, little b. If we want to note that recombination has happened, we just draw the chromosomes and the alleles wherever they go. So if recombination has happened between A and B, and A and B or B, I'm sorry, A and A or B and B have exchanged locations, then we just write it as our new chromosome has big A and little b, and the other chromosome has little a and little b. We're going to see this notation most often from our textbook, but we might see either one. When crossing over occurs, we end up with recombinant chromosomes, and we end up with 4 options. So let's say that we have 2 sets of chromatids, and here we have A and here we have B. And then if these chromosomes switch places, and these chromosomes switch places, we end up with 4 options. We end up with AB, little a, big B, big A, little b, and little a, little b. So there are 4 phenotype offspring, 4 phenotypes in our offspring. And we say that some of them are non-recombinant. These ones on the outside have not undergone recombination. So they're going to have the parental phenotype. And then the other ones are going to be our recombinant gametes. And these are going to produce recombinant phenotypes. So our chromosomes are directly linked to the phenotypes of the individuals. So we've got our parental and our recombinant. And so what this looks like for our offspring is that we have our parental phenotypes, and we can see in those parents what their phenotypes are. So in this case, we've got a fly with a gray body and normal wings, and so we would expect that some of our offspring would resemble that parent. And then we have a fly that has a black body and vestigial or small wings. So we would expect that we have some individuals with that parental phenotype. So we've got the parents on this side, and then we expect our recombinants as well. So that would be this guy here who has the gray body but the vestigial wings, and this guy here who has the black body but the normal wings. And so those are going to be our recombinants. Remember that our gametes are each going to form there's going to be an equal chance of each of these chromosomes or each of these gametes from these chromosomes if they were assorting independently. But if they are not assorting independently, we are going to have an unequal number of offspring. So we are almost always going to have less recombinants than our parents. Our parentals are always going to be the most common, and then our recombinant offspring are going to be less common. And that's because recombination is always happening, but it's not always happening right between these two genes. And so I just realized that I drew those chromosomes wrong. But crossing over is not always happening between those two genes. These alleles should be up here. But we can also have recombination happening down at the bottom. So there might be recombination happening, but it's not always going to affect the two genes in question. So our goal here is to use information about our linked genes to figure out how far apart they are. Now, initially, this was the one and only way that chromosomes could be mapped. Once people realized that genes that were farther apart exhibit more recombination and genes that are closer together exhibited less recombination, they learned that there was a correlation. And so they started using these recombinant phenotypes to help determine the relative distance between two genes. And so in order to do that, we are going to look at what we call a linkage test cross. Now, if we were to work out this test cross as a dihybrid, we would expect to get a 1:1:1:1 ratio. So our linkage test cross is always going to be compared to that 1:1:1:1 ratio. And so if we have to do a chi-squared analysis on there, we're doing the analysis for the linkage test cross. And then in that case, a chi-squared analysis would tell us if these genes were linked or not. So if we have two completely linked genes, meaning they are so close together that recombination never happens, we're only going to get the parental phenotype. If they're really far apart, it is very likely that recombination is going to happen in between them. And then we would still get both types of parents, but we would get an equal number of each type. And so that's where we get our 1:1:1:1. And we would expect that 50% of our offspring are going to be recombinant and 50% of our offspring are going to be parental type. So crossing over can happen anywhere along a gene, although there are some places that are much more likely. So for example, the closer that we get to the centromere, the more tightly compact our DNA is. And when we have super compact DNA, we have less chance of recombination happening. And so the closer we get to the centromeres, and then also the closer we get to the telomeres, the less likely that recombination is going to take place there. But even along the chromosome, there are some places that are more likely than others to recombine. So we can kind of take that into account as we're doing our problems. We'll talk about that a little bit later. We also have the inhibition of one crossing over event that's going to block another. So if we have a crossing over event right there between close to our A, we cannot have another one that is right there on top of it, because the physical distance, the physical space that that crossing over event takes up is going to block anything else from happening too closely. So if anything else is going to happen, it would have to be farther away if we're going to have multiple crossing over events. We usually draw this as just 1 or 2 crossover events just for simplification, but crossing over actually in real life happens at many locations along a chromosome. And then I like this diagram here because it shows the difference between non-crossing over chromosomes. So if nothing happens, if no crossover event happens in between A and B, we're just going to get our parental types. And if crossing over does happen between A and B, you can see that we have these 2 new recombinant chromosomes in the center, which are now new combinations of those traits.